Today we discuss the bacterial cytology. Are there all the parts? This is the bacterial cell. This is the infrastructure of the bacterial cell. We will first we study the cell wall. Why study? Determines in shape, protects cell protection from the toxic substances, site of action for the antibiotics. Location of the cell wall is the outermost and the rigid layer. Types of the types of the cell wall based on the gram staining. So we two types of the cell wall is gram positive cell wall and the gram negative cell wall. The color of the gram positive cell is the purple color and the gram negative is the pink color. Is uh, composed by the Christian Gram in 1884. These are the characters and the gram positive and the gram negative bacterial cell walls. First, number of layers. In gram positive bacteria there is 1 and the gram negative is a 2. Thickness of the gram positive is a thick and the gram negative is the thin. Outer membrane is gram positive is absent and the gram negative is present. Periplasmic space in gram positive is present in the sum and the gram negative present in the all. Chemical composition: peptidoglycan, teichoic acid and the lipopolysaccharide. Lipoteichoic acid is a gram positive and the lipopolysaccharide proteins and the peptidoglycan is gram negative. Purines proteins in gram positive, absent in gram negative, present. Lipid in gram positive, less and the gram negative more. Peptidoglycan in gram positive more and in gram negative less. Permeability of molecule is more penetrable in gram positive and the less penetrable in gram negative. Resistance to the molecule in gram positive is less and the gram negative is the more. This is the cell wall of the gram positive and the gram negative organisms. In gram positive organism is a peptidoglycan layer is present in the more amount, that is a thick layer, and the gram negative organism is the thin layer. In gram negative organism extra outer membrane is present. Peptidoglycan structure in gram positive, that is amino sugars, N-acetyl glucosamine and N-acetyl muramic acid, that is NAC and NAM, proteins and the teichoic acids. Brown's lipoprotein, that is the BLP. Brown's lipoprotein found in the some gram negative cell walls, the most abundant membrane proteins. It is bound by the covalent bond to the peptidoglycan layer and is embedded in the outer membrane by its hydrophobic heat. BLP tightly links the two layers and provides the structural integrity to the outer membrane. Brown's lipoprotein consists of the phospholipids and the lipopolysaccharides. This is the double membrane of the gram negative bacteria. Gram negative cell wall chemical composition is the first is the lipid A, next is the core polysaccharide, third is the O side chain. Functions of the lipopolysaccharide is the give negative charge to the surface, helps in attachment, stabilize the membrane, create a permeability membrane, prevent entry of toxic substances. O side chain protects bacteria. Lipid A is toxic, that is endotoxin, cause the serious septic conditions in the body. Next, outer membrane is more permeable than the due to porins. Porins are proteinic in nature, tube shape, allow passage of molecules smaller than the 600 to 700 D. For larger molecules, carrier proteins are there. Components external to the cell wall. First is the capsule, slime layer and the S layer. Capsule is first is the capsule. Chemical structure of the capsule is the polysaccharide, proteins, polysaccharide, that is the protein. Glyoxy capsule slime, when the layer is well organized and not easily washed out, when it is the zone of diffuse under organized material that is removed easily. This is the capsule under the microscope. This image show the capsule show under the microscope. Functions of capsule: reduces the phagocytosis, storing water, prevents from the desiccation, helps in attachment. S layer external to the cell wall. Functions of S layer: protection against the ions and the pH fluctuation, adverse the surroundings, maintains shape, promotes the adhesion, adds the property of virulence. Next is the pilus or the fimbrium. Short fine hair like the appendages, visible under the electron microscope only. The cell may have the 1000 of pili, slender tube, 3 to 10 nanometer in diameter, several micrometer in the length. This is the structure of the pili. Pillars helically arranged are pillin proteins, that is R, and the chemical composition protein is the L chain. Functions of pili: attachment, rock surface host cells. Some may help in the motility. Example type 4th Jacquerie motility up to several millimeter. Gliding motility, example myxobacteria, and conjugation. Flagella, that is the flagellum, thread-like locomotory organelle extending outward from the CW and the PM. Slender rigid structure to 20 nanometer across and the 15 to 20 micrometer long, stain and can be seen under the compound microscope. Ultrastructure under the electron microscope. Arrangement of the flagella is the monotrichous, amphitrichous, lopotrichous and peritrichous. This is the 4 different structure of the flagella. Ultrastructure of flagella under the TEM. This is the ultrastructure of the flagella is shown in the TEM. Flagella ultrastructure, 3 parts: filaments, basal body and the hook. Next is the ribosomes. Ribosomes, location, cytoplasm and some attachment to the PM complex structure. Composition of the ribosomes is the proteins and the ribonucleic acid, that is RNA. Parts is the 50S and the 30S, and S is the Sedberg unit. Functions of ribosome is the protein synthesis, folding of protein by special proteins called as the chaperone. Ribosome contains size is 14 to 15 nanometer by the 20 nanometer. Molecular weight of the ribosome is 2.7 million. The nucleoid, the structure of nucleoid forms double-stranded DNA, that is deoxyribonucleic acids, linear chromosome. Some have more than one chromosome. A chromosome is a structure of DNA, proteins and RNA found in the cells. It is a single piece of the coiled DNA containing many genes, regulatory elements and other nucleotide sequences. Chromosomes also contain the DNA-bound proteins which serve package of DNA and control its function. DNA encodes most or all of an organism's genetic information. Some species also contain the plasmids or other extra chromosomal genetic elements. Genetic analysis: 60% DNA, 30% RNA, 10% protein. E. coli DNA circle is a 1400 micrometer. Plasmid. Example of the plasmids is the bacteria, fungi, yeast. Small double-stranded DNA molecule exists independent of chromosomes, linear and circular. Few genes, less than 30, not essential for survival. Selective advantages: plasmid is a replicate autonomously, single copy, produce one copy per cell, able to integrate into the chromosomes and gets replicated episomes sometimes lost. The loss of plasmid is the curing. This is the types of the plasmid, their host and their functions. First, fertility factor. Next, metabolic plasmids are next R plasmid. Next is a cold plasmid, and next is a virulence plasmid. Only the metabolic plasmids and the R plasmids is the host, it changes, but all the three is the their host is the E. coli and their function is the conjugation, lactose degradation, symbiosis, nitrogen fixation, resistance to the antibiotic, cholecystine production, enterotoxin, siderophore, cell membrane, retained cytoplasm, selective membrane, prevents the loss of essential components, transport system, waste excretion, protein secretion. Plasma membrane functions: location for the respiration, photosynthesis, synthesis of the lipid and the cell wall constituents, has receptor molecule and to detect and respond to the chemicals in the surrounding, essential for the survival of the bacteria. This is the structure of the cell membrane. Next is a fluid mosaic model. Bilayer phospholipid, amphipathic proteins float within the 5 to 10 nanometer thickness, polar hydrophobic head, long non-polar hydrophobic end. Proteins peripheral 20 to 30%, integral 60 to 80%, no cholesterol but the haploids. Internal structure of the cell membrane is the tubule, vesicles and the lamellae is more important factors. PHB synthesis, when the carbon is excess and used for the biosynthesis and to make ATP. PHB are referred to as the poly P hydroxylenate PHM, glycogen, polymer of glucose, storehouse of the carbon and the energy. Polyphosphate function is the source of the phosphate, use as the source of the phosphate for the nucleic acid and the phospholipid biosynthesis. Note is the phosphate in the environment is the limited. Sulfur oxidize reduce sulfur, that is H2S. Magnetosomes. So what is the magnetosomes? Some bacteria can't orient themselves within the magnetic field because of the magnetosomes. Magnetotaxis. There are the intracellular particles, ion material. Function is not known. Next is the gas vesicle. Planktonic, those live in a floating state because of the gas vesicles. These structures contain the bioency of cell. Example, cyanobacteria, also called as the BJ, that is the bloom. Purple and the green sulfur bacteria, archibacteria. Note, eukaryote don't have this structure, that is the gas vesicle. General structure: spindle shape, filled with the gas, made up of the protein, hollow yet rigid, variable length and the diameter 300 nanometer to 1000 nanometer by the 45 nanometer to 120 nanometer, number few in the 100 per cells. Molecular structure is a major gas vesicle proteins, that is a GVPA, small hydrophobic, and the rigid is 97%, and the minor protein is the GVPC. Other inclusion is the cyanophyllins and the granules. Equal amount of the arginine as aspartic acid store extra amount of the nitrogens. Next is the endospore. Endo is the within, enable cells to endure difficult time, temperatures, trying nutrient depletion, etc. Dormant stage of bacterium used for the dispersal, example bacillus and clostridium. Schematic presentation of the endospore. We see the coat, outer membrane, cortex, germ cell wall, inner membrane, and the core. Endospore formation of the germination. First stage the activation, next stage is the germination, and last stage is outgrowth. Activation, use of the elevated temperature. Germination is specific nutrient like the alanine, and last is the outgrowth. This is the endospore cycle. We see the all three stages. First is activation. Endospores get ready for the germination. Germination, rapid process, spore loss of the refractivity. Outgrowth is a swelling due water up, takes synthesis of the new RNA, proteins, and the DNA. Characteristics: dipocholinic acid in core with the calcium in the core 10% reduces the water content of the core. Endospore become the dehydrated. Increase heat resistance, make cell resistance to the chemicals, keeping the enzymes inactive in a core. pH 1 unit lower than the vegetative cell. High level of the small acid soluble proteins bind tightly to DNA in the core, protection from the UV desiccation and the dry heat. And second is the carbon and the energy source. So guys, we studied all the bacterial cytology. Thank you.